Okay, I think we're ready to go. So welcome everyone. We're so glad that you could be here with us today. We are excited to be hosting this webinar associated with our new OCB working group called Meta-Eukomics. Our working group is performing an intercomparison using micro-eukaryote metatranscriptomes, but we are also more broadly interested in hosting these bi-monthly webinars as a space to discuss omic methods and applications to microbial ecology. So our organizing committee consists of myself, Harriet Alexander, Sarah Hu, and Adrienne Marchetti, who will be joining us a little bit late. So today's webinar theme is omic intercomparisons and metaproteomic applications. We have Mak Saito from Woods Hole Oceanographic Institution and Erin Bertrand from Dalhousie University. Before we introduce them, we have this very quick overview of our working group. And after the talks today, we're going to have a 30-minute breakout discussion for those interested in engaging further about these topics. We'll talk about lessons learned from the metaproteomics intercomparison and considerations for other omic intercomparison efforts. So if you can stick around, we would appreciate that. So I'm going to pass this off to Harriet, and we'll go back and forth for this short presentation.
Thanks, Natalie, and thank you everybody for coming to our first webinar. Yes, great. So we have a recently funded working group through OCB, which we're really excited about. We're largely focused on metatranscriptomic intercomparison with an eye specifically towards eukaryotic organisms. And I think anybody who works within the omics sphere, especially in kind of these larger oceanographic programs, can really appreciate the importance and necessity of starting to understand how well we can compare samples between labs, between groups, between cruises, if we want to actually start developing a more holistic understanding of how our ocean ecosystem works. So broadly speaking, the goal of our program is to run a small-scale intercomparison of eukaryotic metatranscriptomic methods, focusing both on RNA extraction procedure as well as bioinformatic analyses by lab. And Natalie's going to talk through a little bit more on the details of that project. But the outcome of this, we hope, is a white paper or a peer-reviewed journal article focusing on the results of our intercomparison effort, as well as a report on the best practices that we've inferred in eukaryotic metatranscriptomics. However, that's kind of a smaller group, but we really wanted to have a broader kind of community-wide experience, which is why we were hosting these webinars. But community-wide, we want to kind of begin to think about how existing and developing omic tools can be used to further our understanding of marine microbial diversity and function. So even outside of metatranscriptomics, which is why today we're leading off with some metaproteomic work, and kind of better describe the utility and limitations of methods that we currently use, and most importantly, foster exchange between intercomparison efforts so that we aren't totally siloed based on what we're focusing on, such as metatranscriptomics. So next slide.
Yeah, and we can give you an overview of our progress to date. So we started this initiative about a year ago is when we started planning it. We started off with biomass that was collected by Mak Saito's research group on a cruise last May to the Costa Rica Upwelling Dome region. Mac's group concentrated biomass in those filters, sectioned them into slices, and then Harriet then distributed them to participants. And we had about 14 different groups doing this with us. So we came up with this experimental design matrix where groups use different RNA extraction procedures, different choice of library preparation, either poly-A selection or rRNA depletion, and in some cases, different sequencing facilities. So we're calling all of that our wet lab portion of the intercomparison. And just now we got our sequences back and we're starting the informatic portion of this. And so all groups are now starting with the same raw sequence files, but they're free to use the bioinformatic workflow of their choice. And the intent there is to determine the extent of variability just due to bioinformatic choices alone. So we have this wet lab step and this informatic portion of the intercomparison. And that was modeled after the metaproteomic intercomparison that we will hear more about today. We also just wanted to give you an idea of what the metatranscriptomic sampling and analysis process looks like, and then the steps that we are going to consider in our intercomparison. So you can see that there's variability and different decision points built into each of these steps. Since Mac's group collected the filters, we are starting at step 2, which were the extractions. We also had groups, as I mentioned, use different sequencing prep options and different sequencing facilities, different throughput or the number of reads that were generated from that. The informatics will cover assembly decisions, choice of assembler, whether contaminants are removed. We also are going to incorporate annotation decisions, like the type of databases that you're using and how you're doing those annotations. And then downstream steps would be read mapping, differential expression, tools, and choices. And I'll pass this back to Harry.
Sorry. So this is the first of our bi-monthly, every other month webinars, which we have coming down the stream. As Natalie said, today is focused on meta-proteomics and their existing and kind of more advanced or further along down the process intercomparison that they did, as well as applications. But upcoming, we have planned bioinformatic approaches looking at functional and taxonomic assignment methodologies that are used across both metagenomic and metatranscriptomic platforms, methodological considerations such as the addition of standards and extraction methods used, and also a variety, we're hoping to get a variety of people to come and talk about applications of meta-omic approaches to better study microeukaryotes and have a couple of use cases that are shared. And also more to be determined. We sent out a poll earlier on this year looking for interest, ideas, and also nominations/self-nominations for participating as a speaker in these events. So please let us know if you'd like to have any other topics covered, and be in touch. And we will be sharing kind of upcoming schedule with you shortly. But with that, I'm going to pass it off to Sarah, who's going to introduce our first speaker.
Thank you, Natalie and Harriet. So while Mac is getting set up, I get to introduce him today. Dr. Mak Saito is a senior scientist and Stanley Watson Chair in the Marine Chemistry and Geochemistry Department at Woods Hole Oceanographic Institution. And he received his bachelor's from Oberlin College in Biology and Environmental Studies, went on to be a joint program student, and get his PhD in chemical oceanography at MIT at WHOI, and was also a Henry Hess postdoc fellow at Princeton before coming back to WHOI, or at Woods Hole, where he is now. His research is really involved in looking at micronutrient biogeochemistry, trace metal physiology in microbes, marine metaproteomics, metalloenzyme functionality, and he's recently led an OCB marine metaproteomics intercomparison effort, which is very much in line with our working group's interests. And maybe our working group was inspired by that intercomparison effort. And so we're really excited to hear him talk about those efforts today and some of those results. So welcome, Mac.
Great, thanks so much. Yeah, it's really a pleasure to be here. Thank you for the invitation to speak. Yeah, so I'll talk today for about 20 minutes about our ongoing ocean metaproteomic analysis. And this is really the effort of this Ocean Metaproteome Consortium and all of these collaborators here shown. So I just really want to highlight them and thank them for engaging and participating and sending in data. And also I want to acknowledge our advisory committee of Dasha, Mike, and Ben as well. So this project started a few years ago. We had a virtual workshop in 2021, and we recently submitted a manuscript to BioGeoSciences. It's in revision, and that's an open review process, so you can actually go look at it right now. It's in revision. And I want to, of course, acknowledge OCB. This project was an OCB project, a working group project. So let's see. There we go. So maybe you may not be as familiar with metaproteomics, so I'll just briefly talk about that. It's a useful data type that, where we're measuring proteins, which gives us an ability to connect biodiversity with function. And we had a working group a few years ago on data sharing, and we made this graphic of some of the challenges that we face: sample collection, analysis, results and interpretation, and dissemination. And there's a lot of, it's a complex method with a lot of attributes here, and you'll see that in the intercalibration. And I wanted to point out also that there is a Meta-Proteomics initiative a group that launched an intercomparison as well, that was published in Nature Communication a few years ago, led by Tim van der Bosch. And so there's a nice sort of parallel effort in the medical microbiome community. And metaproteomics is starting to mature as a useful science, useful data type. And I just wanted to show one example here from some of Natalie's work with our lab and Harriet's lab, where we did a multi-omic ocean section using Clio, an autonomous vehicle. And I'll talk about that briefly later. But from Bermuda to Woods Hole, about 1,000-kilometer depth section with one station going out to 4,000 meters and able to collect a lot of data. And there's a combination of transcript data and protein data shown here with all kinds of different taxa. This is up on bioRxiv and is in revision now. So just wanted to sort of show that the data is starting to mature as a data type and being useful. I won't go too much into the science of this at this point. So intercalibration is a little bit of a scary effort, as one might imagine, but it's really essential, as Harriet mentioned, for a global research effort where especially if we want to compare data across space and time. And an inspiration for a lot of this is geotraces. We've been part of the geotraces community measuring cobalt for a number of years, and that intercalibration, I think it's not an understatement to say that intercalibration transformed the trace metal community for the better. It really built trust and collegiality, and it laid the foundation for this global program. And this is, this is the ocean sections that Geotraces has completed or is planning to do, and this is their standards and intercalibration committee schematic, where there's a lot of steps in terms of validation of data to prove, to allow this intercomparability across space and time. And so lab intercalibration is one of these. This can also include development of consensus standards and intercalibration using standards for every submitted data set, and then using crossover stations when cruises happen to pass the same point, making sure to sample that, and then checking for oceanographic consistency. And I put this up here because, having gone through this, this is a handwritten note that Ken Bruland, who was one of the sort of godfathers of trace metal chemistry, wrote to me, trying to, herd me as a cat, trying to get me to submit my data for the intercalibration. So it was a nice little hand felt note that certainly raised my blood pressure a little bit, being like, oh geez, Ken Bruland needs me to submit my data. So it's possible, but it really is a good thing. So I just want to emphasize that. And of course, part of the big picture here is, can we assemble data to make something greater than the sum of the parts? And the ongoing biogeoscapes effort is, part of this idea of, can we understand ocean metabolism and nutrient cycles on a changing planet? And intercalibrated data would really be essential to this kind of effort. So that's this, and other projects, global scale projects, are part of the end goal of trying to of all this community work. And if you're interested, there's more information at this website, and you can sign up on the mailing list. So our project was a collaboration between these institutions and participants shown here. This is a screenshot of one of our working group meetings. And our timeline goes way back to 2018, when we collected the samples at the Bermuda Atlantic Time Series Station. We distributed the samples in 2020 and 2021 to interested parties. And then we had our first workshop that fall, where people submitted data, and we started to talk about the data. And then we worked on the report and the manuscript preparation, and just recently submitted the manuscript this past year. And some attributes of the study that are important to know: it was a blinded study, so I still don't know whose samples are whose in the study, and the files were anonymized. So in that sense, it takes some of the pressure off, because you submit the data, but everyone has a 3-digit anonymized number then to work with, and you'll see those numbers. And the samples were collected to Bermuda, a similar design to what Harriet showed. We clamped together 2 McLean pumps, 2 heads each, and deployed them, and pulled up filters that were pretty green. They're from the upper water column, and then we cut them into eighths. So each eighth of a slice had about 21 to 29 liters filtered, depending on which exact filter it was. There were pump heads on meters on each of these. And we sent out the 0.2 micron fraction to laboratories. So it's 0.2 to 3 micron fraction. And then 2 slices were used for metagenome sequencing. So because proteomics relies on mapping to nucleic acid sequences, and that was done by the Naval Research Laboratory, their generous efforts. And then the informatics assembly was done by Jackie Saunders. And some of the informatics samples, so that was the wet lab sample. Some of the informatics samples were also collected by Clio because it was doing its engineering and science trials at Bermuda. And I'll just briefly talk about this because a lot of people are curious about this vehicle. This is a vehicle that collects large volume water filters. You basically drop it from the ship and it can go down to full depth and come up and collect samples with no wire attached. And then it comes up to the surface and sends a text message of its location, and then you can go pick it up, and it has carousels full of 142 millimeter filters that can then be sliced up. And so we use those for the informatics samples. And just for fun, here's a video of its deployment on the most recent Costa Rica Dome and Equatorial Pacific cruise that Eric mentioned as well. Those are the samplers on its side. And there it is, just doing a little testing before it dives off in the distance. Okay, so our study design involved having a wet lab component and an informatic component. We had 9 labs in the wet lab component and 6 in the informatic. The methods were largely, the approach was largely method agnostic, so people could use whatever method they wanted in terms of extraction and mass spec and informatics software pipelines. We did stipulate just a few criteria, and one of them was really that we wanted people to use one-dimensional chromatography. There's more complicated chromatographic methods to get deeper into proteomes, but we figured that might make it more difficult to compare, so we made that a constraint, as well as the total protein injection. We had some suggestions there, and we asked for technical triplet analyses. Let's see. And then we followed this approach. DDA is just a kind of proteomics data-dependent acquisition. So this study was entirely data-dependent acquisition. A future study could be data-independent acquisition, another kind of data type. And we had a first lesson right at the onset where 2 of our 9 wet lab participants had very low protein yields and IDs. Those 2 labs didn't use SDS detergent, like the other 7 labs did, and it seemed to have strongly affected the results. And this might have been just sort of not being used to the sample type. Proteins were embedded. I mean, the microbes in the oligotrophic bats region are really embedded into the filter, and so getting them out with detergent really helps. So we opted to remove those 2 labs, and then a future recommendation is to use detergent, particularly something strong like SDS. And then a second lesson came up immediately too, is that we had this submission of process data. We realized very quickly it was very difficult to submit, to compile this data because it was coming from all these different software packages that use different informatic pipelines, software and statistics and protein inference methods. So it was really comparing a lot of apples and oranges. So we did a little bit of comparison for the paper, but very quickly we moved to just a complete reanalysis of everybody's raw data so that we can have a uniform pipeline to really make this easier. And I think this really highlights a second lesson, is that finding some sort of data output standards to allow a uniform data integration is really important when working with many data sets. And here's some of the results where we're looking at discovered proteins versus unique peptides. And so just for those of you who may not know, all of this is what's known as bottom-up proteomics, where peptides are measured. So that's our base unit, is a piece of a protein that's enzymatically cleaved by the enzyme trypsin, which cuts after arginines and lysines. And so you can see this is just a short protein that's got cleavage sites after every K and R. And so this would be a tryptic peptide right here. And here's another one, some very short ones here and things like that. And so the mass spectrometer is measuring that as our base unit, and then we map them back to longer sequences that the nucleic acids have predicted. And So this is basically our base unit of what's measured, peptides, and then these are the proteins they map to. And so multiple tryptic peptides can map to a protein, and so that's why there's more peptides than proteins. And in general, we saw a few things here. First of all, these are the labs that had low yield, protein yield. So they basically were the two labs that were excluded later. We had a really nice correlation then between discovered peptides and proteins. And so more peptides discovered leads to more proteins. Interestingly, we're not saturating. There's no sort of rarefaction curve that's starting to happen here. More peptides means more proteins, and that'll be a theme you'll see later on in this talk. And part of this was by design. By asking people to do a one-dimensional analysis, people are going to be discovering less peptides, and people are using different machines. So there's some variability here in the number of peptides they discover just by the nature of the approach they used. But we can do much better. Here's an example of a two-dimensional analysis that's a more sophisticated analysis that's getting over 18,000 peptides. And so this is apparent here in what we're calling the long tail of the proteome. So here are 7 labs, and when we compare the proteins in terms of order by abundance for the 8,000 total proteins that were found across the 7 labs, you can see that each lab has some proteins that are more abundant that are discovered. When they're shared between the 7 labs, those are the ones that really are most abundant, right? Or the ones that all 7 labs see are proteins that are most abundant, right? And it's these rare proteins that become much more stochastic in terms of what this data-dependent acquisition method is discovering. And also notably, each one of these individual proteins is less than 1% of the total spectral counts, right? So many proteins adding up to make the proteome, but there's a long tail of sort of undiscovered proteins. And again, that's sort of by design because we ask people to use a one-dimensional method that isn't as deep, but there's obviously a lot more proteins in the sample. And that's part of the challenge in metaproteomics, is that there's a lot of depth and complexity in the sample, and how do we get our machines to measure that, right? But this really brought up a lesson 3 that you'll see a little later, is that it's actually difficult to explain people differences in proteome depth versus this shared core proteome, which is what we've focused on in the paper. And so we can see some of the consistency of metaproteome identifications across laboratories, and this is a Venn diagram of proteins shared between 7 laboratories, and as well as intersection plot. And these are plots made by Maggie Mars Brisbane, thanks to her. And what you can see is that we're seeing about 1,000 proteins shared between the 7 labs, and a number of proteins that are seen in various other parts of the Venn diagram. And similarly, on the peptide level, we see about 1,400 peptides shared between all 7 labs, shown here. And that's out of a total of about 27,000 total unique peptides. So only about 5% of them were shared across the 7 labs, right? And you can see the different combinations of where peptides are discovered. In some labs, we're getting more than 15,000 peptides. So some have deep coverage and some have shallow coverage, just based on the instruments. And then we compared the spectral count, the quantitation across these samples. And we thought we saw what we're interpreting as pretty good consistency on a quantitative level. This is big omic data, thousands of data points. So comparing them and seeing what looked like coherent relationships was exciting to us. And we can do 2-way linear regressions of all of these pairwise comparisons, and you can see that many of the laboratories are getting in the 0.5, 6, 7 range of R² values, and that's shown down here in the mean of the pairwise regressions, right? And here are the total proteins discovered. So we're seeing a lot of shared proteins between labs, and we're seeing quantitative consistency between the labs. And Jackie Saunders did this nice quantitative Sorensen analysis for similarity of the data. When we take the top 1,000 proteins, which is about 75% of all the protein spectral counts, we're seeing roughly 70 to 80% similarity between most lab groups. If you can see that these are pairwise comparisons of the triplicates shown here, and you can see the values there. And then, of course, this is the unison line, so you would expect a lab to be perfect with itself, of course. We can then break this down into taxonomic, phyloproteomic consistency, and you can see that the broad picture is that all 7 lab groups are getting, a similar picture in terms of major taxa showing up. Labs that have deeper proteomes may be getting more slices, up here at the top, versus labs with shallower proteomes. But in general, the taxonomic picture looks pretty uniform. And similarly, on the functional level, in terms of KO groups, we're seeing a lot of similarities. And because this is at the Bermuda Atlantic time series, phosphorus stress is is a big factor there. And so the most abundant protein here, down here at the bottom, is the phosphate transporter belonging to Prochlorococcus in blue. So that was encouraging as well. And then we did an informatic pipeline comparison, and we had 2 North Atlantic samples. These are the ones collected by Clio. We stuck a little Easter egg into the paper here. We're calling them Oceans 8 and Oceans 11 after the movies, because we had to call them something, and why not that? And then let's see. We had participants use 8 software packages, or what are known as peptide to spectrum matching engines. So that's where you're matching the peptides to the spectrum and to the nucleic acid sequences underneath that. And that came from 10 different laboratories. And in general, we were pretty pleased to see pretty consistent results. All of these engines were able to handle the complex metaproteomic data. The spectra are really pretty complicated, but they were all getting in the thousands of hits for both sample sets. And there's a lot of factors inside each software package in terms of the settings too, which may affect the variability here. So it's not necessarily a apples and apples comparison of software packages because of those various settings. But importantly, they all seem to work on metaproteomic data, which was news to some of us. And then for full transparency, I'm going to show some of our reviews, just so people know what the experience is in trying to get intercomparison studies published, right? That Campy paper was published in Nature Communications, which was a great opportunity for them. Our community wanted to go to ISME, and so we tried it, and the intercomparison project was originally just desk rejected off the bat for being out of scope. And so we were then resent to ISME Communications, and we had 2 reviewers, and they really they got hung up on this proteome depth question, and they seemed to miss the shared proteins and peptides that we had focused on. And so, that really sunk us there. So reviewer 2 said also there were large variations in the number of purported proteins identified and apparent abundance. So that kind of sunk the manuscript, and the editors just sent back a form letter rejection. So that was disappointing. But that happens. And so we went to Biogeosciences, which is an EGU journal that has open review, which is, in my opinion, a really fabulous process. And the reviewers are really excellent in terms of their knowledge and positive about the paper, saying the big picture microbial community composition and functions are largely consistent across lab groups. And this is encouraging news, and as detailed in the manuscript, the overall comparability is, despite a wide variety of applied methods, quite high between laboratories. So we just got this last month. Unfortunately, the comment period is closed, so none of you can comment anymore. But normally, you can comment during the open review process. But you're welcome to go read the manuscript there if that's of interest. And so, just to summarize, lessons learned. We had a diversity of approaches allowed that led to some useful observations. The strong detergents turned to be really important for extraction. We also saw a difference in purification methods, beads versus S-traps. That was consistent with some other people found. A pretty small effect, but it's consistent with other studies. We also saw that different software pipelines were really difficult to combine, and that suggests a need for informatics standards. And it was difficult to frame the results for the reviewers, right? And I think we did a better job in the revision that we sent off to Biogeosciences. And then, some future areas we could work on are extraction comparisons, reanalysis of extracts, moving from a sort of a semi-quantitative intercalibration study to intercomparison study to a more quantitative intercalibration study, where we are using new methods that are better at absolute quantitation, such as this DIA independent method I talked about, and the targeted methods, and using reference standards. And I think this ties in well to what Erin will be talking about shortly as well. So I'll end there and just acknowledge again this community of people who came together to work on this, and the project team, Matt, Jackie, myself, and our advisory committee, our colleagues who help with the Meta-Eukomics, OCB and National Science Foundation for funding, the OCB office, which has been incredible for helping us throughout the whole way, and the BATS field team as well. Thanks very much.
Wonderful. Thank you so much, Mac, for that overview. We can take a few questions now. So if you have one, you can use the raise hand function, or you can drop your question in the chat. We can take a few. If there are many, we can also take them after Erin's talk. Okay, I can get us started. So Mac, I was interested in that detergent finding. Could you explain a little bit what you think is going on there? So were other groups not using detergent? Were they still extracting cells? How might they have been doing that? Yeah, we're interested in that for our intercomparison, just how those different procedures might change the RNA pool that we end up with.
Right. Yeah, I think some groups tried to do this by sort of buffer and lysis, and I think another group used a weaker detergent, sort of one of the designer detergents, and as a result, I think because the oligotrophic samples have so many small cells that are sort of really impacted into the filter, they just didn't come off very well with those methods. Whereas, the SDS and heat 95 degrees kind of treatment just does a wonderful job of both lysing the cells and then solubilizing all the proteins. So, and we sent 2 filters to every lab so that if they found that there was a problem, initially they could loop back around. But I think those groups, for whatever reason, didn't do that, and so. But I think that was really a really useful lesson for us actually to see, and for them, I've heard through the grapevine.
Absolutely. All right. So, any other questions from the audience now? Okay. If not, and if you think of one later for Mac, we can definitely ask him. So I'll turn it over to Sarah to introduce Erin.
Okay, thank you, Mac. So Dr. Erin Bertrand is an associate professor and Tier 2 Canada Research Chair in the Department of Biology at Dalhousie University in Halifax, Canada. She received her bachelor's in chemistry and environmental studies from Bates College and was also a WHOI MIT joint program student and got her PhD in chemical oceanography. She went on to do a postdoc at JCVI, the J. Craig Ventnor Institute, and Scripps Institute of Oceanography in San Diego before she began her position at Dalhousie. Her research focuses on looking at trace metal biogeochemistry, vitamin quantification, and metabolism, the polar phytoplankton ecophysiology, and targeted metaproteomics. She is also a participant in the OCB Meta-Proteomics intercomparison work, and so we're excited to hear from Erin, and thank you for talking with us today.
Thank you, Sarah, and thanks all of you for the invitation to share some of our work with you today. It's exciting to follow Mac. Mac was my PhD supervisor. And so you'll see some common threads, of course, in what we're talking about here. But I wanted to share some of the ways that my group's been thinking about using metaproteomic data to help us understand particularly rates of processes. And for this group, I decided to focus on eukaryotic processes through metaproteomics. And so that's the lens I'll share with you today, but I just want to emphasize my group benefited tremendously from participation in the Metaproteome intercomparison project in terms of optimizing our methods and thinking things through as a community. So I'm really excited to see what comes about from the metatranscriptome intercomparison project. So the way that we've been, see if I can get my screen to go forward. Sorry. This was working before, of course. I'm just going to stop my share for a second and try again.
You might be able to try sharing your whole desktop. Sometimes that works for me instead of just the PowerPoint.
Yeah, thanks. Where are we? Okay.
I use the down button for forwarding instead of the right button too. I hope that helps.
How's this? Oh gosh, come on. It's not working again either. Try one more thing, sorry. Okay, there we go. So we've been thinking about 3 different ways to try to get from metaproteomic data to rates. And one is through this combination of cellular protein allocation modeling, whole metaproteome data, combined with key environmental data. We've also been thinking about ways to use quantification of key individual proteins or quantification of pools of proteins. I want to make the point now that all of these are sensitive to metatranscriptome methods choices. And so I'll be really interested to follow through on what you all learn in the intercomparison and translate that to some of our metaproteomic interpretations. But today, so I'll briefly just mention that the work that we've done using cellular protein allocation modeling is exemplified in this paper by Scott McCain from his PhD work in my lab, and where we took this model that Scott made of the diatom Fragilariapsis cylindris. We combined that with metaproteomic data from the Southern Ocean and some environmental data. And we're able to output inferred growth rates and inferred iron acquisition rates from that combination. So I'm not going to talk further about this, but I refer you to Scott's paper if you're interested in this. So for the rest of the time, I'm going to talk about quantification of key individual proteins, such as rubisco, as quantification of pools of proteins, such as ribosomes, towards our efforts to see if we can learn something about rates from metaproteomic data. I just want to mention that we were inspired by one of Mac's papers where he used measurements of NXR from the Pacific to approximate rates of NXR activity in the ocean, and I thought this was really exciting, and started thinking more about what other kinds of processes we might be able to do something similar for. And I've been thinking that this requires a few criteria to be met. We have to be able to make some assumptions about whether an enzyme is process limiting. If we want to try doing this, we need to be able to measure absolutely the concentrations of those enzymes of interest and know who made them. And we also need to know something about the enzyme kinetics and be able to make some good assumptions about their status with respect to reactants and products if we want to try doing this. So a place that I wanted to start was with rubisco. We know a fair amount about this enzyme. It's key in carbon fixation, of course. We know it's an inefficient enzyme. It has low turnover rates. We know that it's often rate limiting. They're often the rate limiting step in the Calvin cycle, and it's often CO2 saturated. So based on this information, we hypothesized that under certain conditions, rubisco concentrations could be used to estimate carbon fixation rates. So then we wanted to move on to the second requirement. Can we make good measurements of rubisco and who made it in the environment? And to do that, my student, Megan Roberts, led this effort to design a series of peptides that we can quantify using a different kind of mass spectrometry than Mac was talking about earlier, where we add isotopically labeled internal standards of peptides, and we measure the absolute concentration of these specific peptides rather than trying to identify a suite of all the peptides present in a sample. So you can see on this screen, we've identified some peptides. We call this the S peptide. This one is broadly found within most eukaryotic rubisco sequences. And then we've identified a series of other peptides that we think are representative of specific groups of rubisco sequences. 3 different peptides that help us represent diatoms, for instance, a haptophyte peptide that mainly targets Phaeocystis, and then others such as this one targeting Micromonas here. And so Megan applied this suite of peptides to measuring rubisco concentrations in the high Arctic. This is in the Canadian Arctic archipelago between Baffin and Ellesmere, or between Devon and Ellesmere Islands here near Baffin Bay. And what Megan was able to show in this depth profile over here, we're showing you the concentrations of this broad eukaryotic rubisco peptide as well as other group-specific eukaryotic rubisco peptides with a CTD profile showing you chlorophyll trace and nutrient concentrations. And so this was encouraging to us. It suggested these rubisco distributions were similar to what we would have expected from first principles, so that was nice. We wanted to know whether the community composition that we were capturing through our rubisco peptides was similar to other metrics for community composition. So are we getting what we expect from rubisco here? And so just comparing to 16S chloroplast-based estimates of this community composition to our rubisco-based estimates, we can see this is broadly coherent. So the rubisco seems to be coming from who we expect it to be coming from in this system. And I'll refer you to Megan's paper, which is out now in limnology and oceanography for more details on that. So we think we're capturing the rubisco from the community members that are important, at least in this region. So then we wanted to turn to our list of requirements. What do we know about the enzyme kinetics of rubisco? And can we make some assumptions that will help us move from these concentrations into potential activity? And here we know that if this enzyme is CO2 saturated, we can use the specific activity of the enzyme to estimate the rate of carbon fixation from the concentration of the enzyme and that specific activity. But what do we know? And of course, some conversion factors here, but what do we really know about rubisco specific activity? I'm showing you here rubisco specific activity versus temperature, and this encompasses most of what we know about rubisco activity. There are large data gaps, I'll emphasize that, but what we do see is that at low temperature, there's low variability. As we get higher to higher temperatures, we see across different groups of organisms, there's a wider range in that catalytic in that activity. So when we're at low temperatures, maybe we can predict this reasonably. So our hypothesis is that at low temperatures, we can apply a small range of kcat values to estimate carbon fixation from rubisco concentration measurements. And so I'm going to show you two examples where we're able to compare our rubisco-based estimates to other kinds of estimates for carbon fixation. This is an example of a collaboration with Jody Young from the University of Washington, where Jody's group collected samples from sea ice and from the water around the West Antarctic Peninsula. And I'm showing you here a comparison in the potential rubisco carbon fixation rates versus C14 measured net bottle production estimates. And what you can see here is that in the water column measurements, which are shown in green, we see a pretty reasonable coherence, and things get interesting and messy when we turn to sea ice, but this is perhaps unexpected in large part because we expect this production in sea ice to be light limited. So lots to explore there, but this was encouraging for us, suggested we're not crazy for thinking that this might be an interesting avenue to explore. Moving forward to the Labrador Sea, which is between Newfoundland and Greenland, we were able to get a lot of samples during a Phaeocystis bloom in 2022. I'm showing you here a time series of satellite images showing the progression of the bloom here. It was massive in April, and then we can see the bloom decline through May. I'm showing you, so we had samples through mid-bloom, through the bloom decline, and then termination, we think. And then through combining two different expeditions, we were able to acquire a number of samples to try to use our rubisco-based measurements to understand productivity. And so this work was led by a PhD student in my group, Scott Polera, shown here. So I'm showing you Scott's rubisco concentrations during the mid-bloom. And I'll just note that the axis is different here. These concentrations are much higher during mid-bloom than our other samples here during bloom decline and termination. And we're showing you Phaeocystis rubisco in brown, diatom in black, and Micromonas in gray there. So this is heavily dominated by Phaeocystis rubisco, which is exciting. So if we apply some similar assumptions and compare our rubisco-based estimates of of carbon fixation to C13-based estimates of bottle production. Here, again, we see reasonable coherence between these and some interesting differences. So we're excited about exploring those differences and trying to understand that a little bit better. But again, we think this is pretty exciting that we can, based on protein concentrations, predict rates of primary productivity. And we're excited about trying to apportion this to different groups of plankton, but we have a bit of a hurdle. I wanted to explain here that right now what we're measuring as the sum of our group-specific peptides that measure each of these different individual groups of rubisco sequences, so phaeocystis, micromonas, and diatoms, shown in these bars here compared to our general eukaryotic conserved rubisco peptide. Sometimes they match really well. Other times they do not. And so we have some work to do to understand this mismatch a little bit more before we can move forward to confidently calculate taxon-specific rates of primary production from these. So might there be analytical issues that we need to think about here? Are there missing groups of plankton that we need to be quantifying? We're not sure. We think that's probably not the case based on other estimates of community composition. But are we missing rubisco diversity within these groups? Probably. And maybe there's something interesting going on with the rubisco complex there that we're not fully capturing. So a little bit more work to do there, but we're really excited about this prospect. So our next steps, we hope to reconcile these differences between total and group-specific rubisco concentrations. That could entail metatranscriptome data mining and culture comparisons, importantly. We're also interested in getting more coverage of our rubisco peptides. We need to do some culture-based assessments. I tend to always want to jump right to the metaproteomic to understand what's going on in the environment, but there's of course a lot that we need to investigate within cultures. And I'm really interested in this time integration. So what kind of time stamp comes with this rubisco concentration? What time interval does it tell us about productivity over? That's a question we don't really know the answer to just yet. So lots to explore there. I want to tell you quickly one last story here about potentially using groups of proteins and quantification of groups of proteins to help us understand rates in these organisms. And I'm going to focus on ribosomes. So as just a reminder, ribosomes are protein RNA complexes that synthesize protein. And we know from classic literature that ribosomal abundance correlates with growth rate in some organisms. And a representation of some of this classic data is shown here on the right. And this is known as the growth rate hypothesis. So we're hypothesizing here that we can use metaproteome-derived ribosomal investment metrics to tell us something useful about taxon-specific growth rates or differences between growth rates, for instance. So this is, we have a lot more work to do in this realm, but we're really intrigued by the potential here. So what the way that this works is that we take our untargeted global metaproteomic mass spectrometry data, like what Mac was describing to all of us earlier, which comes in the form of relative abundance data. We use an annotated metatranscriptome database, and then we can do a bunch of data, interesting data processing steps that I'm really glossing over here today, but we take the sum of the diatom assigned ribosome peptide abundance and divide that by the sum of the total diatom peptide abundance that we identify, and we can compute from that diatom ribosomal mass fraction. And this was work led by Scott McCain, who was a PhD student in my group. And then he applied this to a bunch of different organisms within a metaproteomic data set from the Southern Ocean. I'm just showing you here on the right a representation of what he found for different bacterial groups, just as an example. But this is ribosomal protein mass fraction on the y-axis for these different groups across a time series from week 1 to week 4, where week 4, we know the phytoplankton in that community were limited, were micronutrient limited, and week 1, they were replete for micronutrients. So some interesting observations to me were that there's some things that make sense here. So gamma proteobacteria, which tend to be more copiotrophs, are investing more in ribosomes than, for instance, SAR11, which is a slower grower, of course. And we see this trend in gamma proteobacterial ribosomal protein mass fraction allocation decreasing as so
I want to tell you about how we've been working to use quantification of pools of proteins through metaproteomics to estimate taxon-specific rates using ribosomes. So ribosomes are protein RNA complexes, of course, that synthesize protein. And the growth rate hypothesis states that ribosomal abundance correlates with growth rate in some organisms. Some of the foundational data for that hypothesis is shown here on the right, where we've got growth rate on the x-axis, and we've got RNA content per cell on the y-axis. So this this leads us to the hypothesis that metaproteome-derived estimates of ribosomal investment can tell us something useful about taxon-specific growth rates. So since we can use mass spectrometry data to apportion metaproteomes into protein invested in ribosomes in different taxa, perhaps we can use this to tell us something about growth rates in situ, or perhaps we can use it to tell us something about important ecological traits of microbes. So to illustrate this a bit better, to do this we use untargeted global mass spectrometry data, and we annotate that using metatranscriptomes. And then this is something that Scott McKean put forward in his PhD thesis. We can use the sum of all peptides identified as diatoms and ribosome, for instance, relative to the sum of all peptides identified as diatoms and their abundances to calculate a diatom ribosomal mass fraction, for example. So this represents the amount of protein in a sample that the diatoms broadly invest in ribosomes in that environment. So to illustrate this a little bit more, this is an example from Scott's thesis where he's showing ribosomal protein mass fraction on the y-axis from different bacterial groups on the x-axis here. And what you can see, this is over a 4-week time period in the Southern Ocean, a time series. And what you can see is over that 4-week time series, SAR11 showed low ribosomal investment and invariant ribosomal investment, perhaps consistent with what we know about this organism, that it does not display large changes in function as in response to environmental changes, and it also we know it to be a generally slow grower. This is in contrast to gamma proteobacteria, many of which we know to be copiotrophs that can respond quickly to changes in their environment and can have high growth rates. We see a decreasing ribosomal investment, perhaps reflecting decreased growth rates in this of this group in these samples, and this is consistent with some other ancillary measurements we made about bacterial dynamics in this system. So to focus a little bit more on a eukaryotic example, I'm going to take you up to the Arctic now, where we did a series of experiments to examine ribosomal investment in by diatoms in the high Arctic, in what we think is likely a nitrogen-limited system, where we were interested to better understand the influence of glaciers on phytoplankton. And this was work that was led by Patrick White when he was a master's student in Maya Bhatia's lab, and now Patrick is a PhD student in my lab, where he's continuing to work on these experiments. And what Patrick did was introduce deep water and glacial meltwater in varying proportions to surface ocean phytoplankton communities and examine their proteomic response. This was in collaboration with Maggie, Ariana, and Harriet in Harriet's lab. So this chlorophyll data shows you what happened, broadly speaking, in a bulk sense in our experiment. So mean chlorophyll concentrations on the y-axis and time on the x here. There are a number of different treatments, but really 3 things for you to worry about here. First is that these orange treatments here received little to no nitrogen. The blue treatments received moderate amount of nitrogen, and the purple treatments received much more nitrogen. This was what we expected to be a nitrogen-limited system, so these results make good sense to us. So what we did was analyze the proteome components after 24 hours. So the question is, how did diatom ribosomal mass fraction respond to these additions different meltwater, these different meltwater and upwelling simulations that we think are analogous to how glaciers interact with the surface ocean. So what you can see is that along a trend of increasing nitrate concentrations on the x-axis here, we see an increase in ribosome mass fraction, so an increase in the amount of the proteome that the diatoms invest in ribosomes when they are experiencing enhanced nitrogen additions. This is just after 24 hours in near zero temperature conditions, before we were able to resolve any increase in chlorophyll. So we're really intrigued and excited by these results, and we think it could be a good indication that that changes in ribosomal investment does indeed reflect changes in growth rate. And there might be many other lessons that we can learn from an ecological standpoint about by continuing to interrogate ribosomal mass fraction in different situations. So with that, I'll just conclude by summarizing that I think I've shown you three ways by which my group has been trying to estimate taxon-specific rates from metaproteomic data. The first being using a combination of protein allocation modeling, whole proteomes, and environmental measurements. Second, by quantifying key individual proteins in very specific circumstances when we think we have good hypotheses about what the rate limiting step in a process might be, and sufficient understanding of the enzyme properties. And third, by quantifying pools of proteins, and I've used ribosomes as an example here, so I'm showing you that maybe ribosomal investment can tell us about group-specific growth rates or changes to growth rate. And I want to take a moment just to emphasize that I think we need specific in-depth examinations of how decision-making and metatranscriptome data handling actually impacts all of these approaches, because we lean heavily on metatranscriptome data to do many of the things that we do here. With that, I'd like to thank all my collaborators for what they've contributed to this work. Large team of people working in the Labrador Sea, and then work in the Canadian High Arctic, really led by Maya Bhatia, who we sadly lost this year. On the protein work that we presented from Jones Sound from the Canadian High Arctic would never have happened without Maya's leadership. So I thank everyone for your attention, and I'll be happy to continue discussions as your working group moves forward. Thanks, everyone.